Hello, I'm Stefan Przyborski. I'm Professor of Cell Technology at Durham University, and it is a pleasure to give this year's Charter lecture for the Royal Society of Biology. I'm going to be speaking about bioengineering human tissues in the laboratory. So for many years now, I've had a research interest in studying the differentiation of cells and tissues, and this slide summarizes some of our early work where we were collaborating with other groups in the UK to characterize the first human embryonic stem cell lineage. So these cells have the potential to produce all different differentiated tissue types in the human body. And we test this by transplanting these cells into immune deficient animals and allowing a teratoma tumor to form. However, we want to replace the use of animals in this process, and we want to be able to generate such tissues in a controllable manner, reproducibly in the cell culture laboratory. But unfortunately, conventional two-dimensional cell culture models do not enable this to occur. Cell culture itself, though, has enabled many important conceptual advances. It's particularly useful as a reductionist approach to study mechanisms and address specific questions. The in vitro environment, however, differs considerably from the complexities of real tissue structure and function. And that's an important feature to remember, and we'll come on to that more in a moment. Animal models provide a more definitive test in that cells can be studied in a systemic, physiologically relevant environment. However, the conclusions from animal work does not always extrapolate to the human condition. And of course, there are obvious ethical and moral issues surrounding the use of animals in research. So advanced cell culture technologies provide new strategies for overcoming some of these limitations. And we're particularly interested in the microenvironment in which cells grow within the in vitro situation. This diagram here shows very simply a cell which is receiving a variety of different signals from its environment. And these can be things like changes in oxygen concentration, or the addition of a hormone or a growth factor to the culture medium. Each can influence the behavior of the cell and thus the experimental outcome. And we are particularly interested in the three-dimensional space in which cells occupy when growing in vitro. So in vivo, in real tissue, cells obviously grow in 3D with their neighbours and other cell types, and the extracellular matrix. But more often than not, we pull these relationships apart and we culture cells in a very simplistic environment, such as the Petri dish or the cell culture flask. And when cells come into contact with this foreign substrate, so this is the base here of, say, the Petri dish, you can see they undergo a transition where they transform from a three-dimensional shape to something which is adapted and flattened, as seen in this image. As cells go through this process, they remodel their internal cytoskeleton, which in turn can affect gene transcription and protein translation. So we are interested in developing technology to prevent that shape change from occurring and to recreate a more natural three-dimensional environment which more closely simulates real tissue. And if we can do that, we will enable cells to acquire a more natural three-dimensional architecture. There is evidence of increased cell viability, enhanced structure and function. Such models will be more representative of real tissues and lead to improved predictive accuracy. So this is important in several ways and has significant impact. It can advance research and discovery. Having improved cell-based assays can lead to improving efficiencies in terms of saving time and money in the long run. And of course, in particular, for using and developing models in human cell systems, we can replace and reduce the use of animals. So we set about to develop a material to enable routine three-dimensional cell culture in vitro. And this started with interdisciplinary research to develop the material in collaboration with chemists and engineers. And our aim was to produce a technology that must have the following attributes. The material was inert and did not degrade and remained stable throughout the experiment, so it wasn't an additional variable if it was to degrade. The material was actually the same as an existing 2D cell culture product. So petri dishes and flasks are made of polystyrene. So the material we developed is also made of polystyrene, and we've changed its configuration, and I will show you in the next slide. So the technology must adapt to existing cell culture formats and is also a consumable product. So what we've developed is an inert polystyrene scaffold and re-engineered that into a membrane. If we look along this top line here, we can see the standard conventional cell culture situation where cells grow as monolayers in 2 dimensions. We've changed the geometry of the polystyrene to create this 3-dimensional material, introducing now the Z-axis, and this creates headroom and space in which the cells can now grow in a 3-dimensional configuration with each other, as you see in this image here. These images at the bottom show the scaffold in transverse section at low and high magnification. The scaffold is 200 microns thick. It is 90% porous, so 90% of the volume of the material is actually space, only 10% plastic. And inside it, cells can form more in vivo-like structures, and also on the surface of the material as well. So it's very important to commercialize this technology to maximize its impact, especially getting the technology out into the scientific community for others to use. And this was achieved using a commercial vehicle, a spin-out company from Durham University. So I founded a company called Reinnovate initially to commercialize and launch this scaffolding technology. And it was branded Alvatex. And Alvatex, you can see, is this disc, white disc in the bottom of the wells of these multi-well plates. And there are different formats ranging from 12 to 384 well. And we also have well inserts as well. So this technology has been on the market now for several years. The number of users has increased significantly. There are now well over 100 independently published articles using Alvatex technology. And we subsequently sold the spin-out company to Reprocel, which continues to develop this technology further. So, as I mentioned, substrate determines cell shape and form. So does the technology work? Does it prevent that shape change from occurring? So in 2D, cells flatten, as I showed you before, and the nucleus also flattens. In the Alvatex scaffold, the cells retain a 3-dimensional shape and form, as does the nucleus. So we can see this in action with a simple monoculture system. So this is a single cell type growing on the material. And these are hepatocytes, and they have been stained with fluorescent dye. You can see them growing in 2D here, and they're flattened. Whereas when seeded at low density, so you can see their profiles, the cells retain a 3-dimensional shape when grown in the Alvatex scaffold. And just like real tissue, where you would fix and embed and section and perform histology, you can do the same with this technology. And you see histological images here of the 3D cultures, image also by scanning electron microscopy. On this slide, we're comparing three situations: the growth of hepatocytes in 2D, in 3D, using the Alphatec scaffold, and the real liver tissue. And we do this for all of the models that we develop. We always compare the 3D model with the real tissue because we're ultimately trying to recapitulate the structure of those cells within that tissue. And you can see quite clearly the cells growing in 3 dimensions, resembling a growth structure similar to their native counterparts. But what's really interesting is when one looks more closely at the junctions between the cells, and we see the formation of biocannuliculi between the cells in our 3-dimensional models, which we do not see in conventional 2D culture. And you can see them very nicely stained here with a P-glycoprotein marker compared to real tissue. Cell viability is also significantly enhanced as a consequence of cell growth in the 3D system. And this is data showing for rat primary hepatocytes, and primary hepatocytes are valuable. And we see that this live dying dead assay demonstrates enhanced viability in 3D compared to 2D. And also in the simple system, we see an enhancement in function of these cells, whereby the hepatocytes, these are HepG2 cell lines, are producing high levels of albumin and urea in 3D compared to 2D, and all these data of course are normalized to take into account differences in cell numbers. But also the activity of phase 2 drug metabolizing enzymes is different in these HepG2 cells as a consequence of their growth within the 3-dimensional environment. And this therefore adds more value to the HepG2 system when using this technology. So that's the technology as a basic technique, i.e. 3D cell culture. But what we really want to do is apply it to bioengineer human tissues. And this comes back to a fundamental principle in anatomy and physiology, where structure is related to function. Da Vinci himself reported this in the 1400s, and one of our older learned societies, the Anatomical Society, has this as its strapline. So ex conformationis usus means from structure comes function. So here we're looking at bioengineering complex epithelial tissues using this membrane technology. So an epithelium is defined by a layer of cells sitting upon a basement membrane, and it's supported by its stromal tissues underneath. And the epithelia in the body have different structures and functions depending upon their location and their purpose. So for example, in the intestine we have a simple columnar epithelium suited for absorption and secretion, whereas the skin is stratified and keratinized, offering protection and acting as a waterproof barrier. So the anatomy of these epithelia is very different, as is their physiology and their function. And we can use this technology to bioengineer these structures. So I'll start first by talking about skin. So here's human skin. This is the epidermis and the dermis. So it's quite a simple configuration, but not to belittle the complexities within these different tissue layers. So how do we construct skin? So we use the membrane technology to first culture the primary dermal fibroblasts for a period of time, up to 4 to 5 weeks. And during that time, these fibroblasts are very busy producing extracellular matrix. This is a fibroblast inside our vertex producing lots of collagen, as you can see here very clearly, and it's stained by these immunomarkers. And that builds up over time, and that creates a foundation upon which to build the epidermis. And we see very clearly now the epidermis, which is now on the surface of that dermal compartment, and that has differentiated into its component parts when grown at the air-liquid interface. We compare the model that we've produced to real human skin, and we do that over a variety of different markers, as you can see here, and I won't go through all these data, but the point is the expression patterns of those proteins in the model resemble very closely the expression of their native counterparts in real skin. We can increase the complexities of these models still further, and here what we've done is we've included into the model, you can see with the S100 staining, these are melanocytes, and these are located in the basal layer of the epidermis, and these create a melanin, and that gives you pigmentation of the skin, which you can see very clearly. These are the gross photographs of the skin models, and you see these are darker in the pigmented with the inclusion of the melanocytes. If we look more closely, you can see how the melanocytes are cleverly mimicking the features of real pigmented skin. So in real skin, you can see over the nucleus of a keratinocyte a supranuclear cap of melanin. So these are the melanosomes. And what happens, it is understood that the melanocytes, which you can see here, these dendritic cells, are giving up these vesicles of melanin and transferring to the other cells and producing the supranuclear caps. And that is what is happening within our model. So the model also provides, therefore, a useful system to study the cell biology and the process by which that happens. We can also take the technology and in our skin model and deliberately seed into it melanoma cells. And we can study how these cancerous cells then invade through our tissue model of human skin. And we can compare that closely to real melanoma pathology to examine the similarities of this process, and then use this as a model to perhaps look at the effect of drugs which might influence, say, cell invasion. We can also use this technology to simulate the aging process in human full thickness skin. So as we age, our complexion and skin changes, the epidermis in certain areas of the body thins, we lose collagen from our dermal compartment, and of course there are many products available on the market to suppress that change from happening. So we can use our technology to help in the development of such skincare products, and we can simulate the aging process. So here we see a model of young skin versus aging skin, where we've used cells from a 50-year-old donor individual, and we see a much thinner epidermis, and there is less collagen in the dermal compartment. So let's change tack and look at a different model. And this is the human intestinal mucosa. So in this situation, we have a simple columnar epithelium residing on top of a basement membrane and supporting stromal cells. So this is a single layer, and it is involved in secretion and absorption. Now, the industrial standard of studying this process of secretion and absorption of ions across this epithelium involves the use of a cell line called CACO2, most often grown as a monolayer on a transwell membrane. So a very simplistic system. The structure does not resemble real tissue, and it is well known that the functional characteristics of these cells are different from the tissue. So what we propose is that we can apply our technology to these cells to put them in an alternative growth environment to change their structure and their function and improve their value. So we follow a similar principle as with the skin. We take the alvatex and we fill it with fibroblasts, which produce extracellular matrix. We then seed onto the surface the CACO2 cells, and notice what happens. The cells are now columnar as opposed to flattened in 2D, and they have a prominent brush border, and they're polarized just like their native counterparts. We can maintain these cultures for up to 35 days and longer. We see nice reproducible examples here. We can use various electron microscopic techniques to evaluate their structure. So this is a transverse section of the stromal layer showing the fibroblasts here and here. This is the foundation upon which we seed the CACO2 cells. We're now looking at them from above, seeing the cobblestone morphology, and high magnification shows the microvilli and the glycocalyx. And transmission electron micrograph shows those microvilli very clearly on the apical membrane with nice tight junctional complexes between the cells. And these CACO2 cells produce a nice trilaminar basement membrane at the interface between the fibroblasts and the epithelium. We compare again with the model and real tissue, and we show very clearly that there are similarities in levels of protein expression in the right areas as one would expect. We also look at the function of these constructs. We take the construct, we put it into a chamber system called a Ushing's chamber, and using amplifiers we can record the passage of ions across this human intestinal model. We show that the trans epithelial electrical resistance is significantly lower in our model compared to the existing 2D transwell system, which is much more towards the in vivo situation. We also show that drugs are transported differently and more appropriately in a three-dimensional tissue model system compared to the existing two-dimensional transwell model. We can also take our technology and mimic certain diseases. So inflammatory bowel disease, such as ulcerative colitis and Crohn's, is affecting large numbers of our population. We see 11% of the world's population affected by these types of diseases, and it's increasing. And what we see is the invasion of large numbers of immune cells into the tissue and causing disruption to its structure. So we can simulate this process by modeling inflammatory bowel diseases in vitro, and we can do that by taking populations of macrophages in culture and putting them into the stromal compartment of our model, and we hypothesize that if we stimulate those cells to release cytokines, this will induce phenotypic changes reflecting the disease. So we've done this. We can see very nicely the immune cells within the model. These are the rounder cells. We can use this antigen marker here, CD68, to be able to locate them. And again, we compare our model to real, in this case, pathological tissue. So if we stimulate those immune cells within our model with LPS, we can induce the release of cytokines, which in turn affects the permeability of the epithelium, thus simulating aspects of the disease. So you see a decrease in the resistance across the epithelium and an increase in the permeability of certain markers, such as FD4. So the human intestinal equivalents have a potential not only to study the effect of various compounds and drugs working across that epithelium, but also to simulate disease. We can increase its complexity still further by introducing goblet cells, as we have done as well. So to finish, and to bring ourselves full circle, I want to introduce another technology, just very quickly on this single slide, about how now we've used the technology to replace and reduce the use of animals in research. So I started off talking about the teratoma xenograft assay and how we can produce these differentiated human tissues in an animal. Well, we have now developed a replacement, an alternative, whereby we can apply the technology I've described to you today in order to create sophisticated teratoma-like structures in vitro using the Alvatex technique. And this can reduce technical burden. It provides greater versatility because it's controllable. You can add additional morphogens if you so wished and see differentiation on the tissue. It's a simple, much more routine technique and easier to use than the xenograft assay. It's more cost effective, and most importantly, it enables reduction and replacement of the use of animals. So in summary, the environment in which cells grow plays an important role in their structure and function, and the cells do respond very much to their surroundings, influencing their anatomy and their physiology. And I've spoken today about technology developments that are enabling us to create now more sophisticated model systems, the formation of tissue models, in particular human tissue models, that would be beneficial to research, to increase the efficiency of the discovery process and reduce animal usage. So I'd like to finish there, and I'd like to thank you for your attention, and I'd be very happy to take any questions. Thank you.